All right, let's get into a wild story. It's all about how a couple of scientists peeked inside an ancient bacterial immune system and ended up creating one of the most revolutionary tools in the history of biology. We're talking about literally molecular scissors that can hack the code of life itself.
I mean, just think about that question for a second. It sounds like something straight out of a sci-fi movie, right? But what if you could actually go into the massive book of life with its 3 billion letters and fix a single typo? That's the kind of power we're talking about here. And the key to it all was found in the most unexpected place.
So it all starts with this thing called CRISPR. You gotta understand, for billions of years, bacteria have been in a constant war with viruses. It's an epic, ongoing battle. And to survive, bacteria came up with this brilliant defense system. When a virus attacks, the bacterium grabs a little piece of the virus's DNA and weaves it into its own genome. It's basically creating a most wanted poster. So if that same virus ever dares to show its face again, the bacterium is armed and ready. Okay, so scientists knew this system existed. They could see the most wanted gallery. But the big question, the real mystery was, how? How exactly did the bacterium use this library of viral mugshots to hunt down and destroy an invader with such pinpoint accuracy? Nobody knew the full story.
And this is the exact puzzle that a game-changing 2012 paper set out to solve. We're talking about the work of Martin Ginnick, Jennifer Doudna, Emmanuel Charpentier, and their teams. They were basically detectives on a molecular crime scene, trying to piece together how this natural search-and-destroy mission actually went down.
So to solve the mystery, they had to line up all the suspects. You know, the key molecules that make the whole thing work. And it turned out there were 3 crucial players involved in this operation.
First up, we have the enforcer, a big protein called Cas9. The team suspected this was the muscle of the operation, the actual scissors responsible for cutting up the enemy DNA. But, you know, a weapon is pretty useless if you can't aim it.
And that's where our second player comes in, the guide. This is a little piece of RNA called CRISPR-RNA, or crisRNA for short. This molecule is made from that most wanted list, and its whole job is to carry the mugshot, the genetic signature, of the virus they need to take out. And then there was the third suspect, the hidden accomplice. This was another mysterious RNA molecule called tracrRNA. Now, scientists knew it was hanging around and was somehow involved, but its exact role? That was a complete mystery. And it turns out, this little molecule was the key to unlocking the entire puzzle.
Okay, so the first huge breakthrough in the paper came when the team finally figured out how these 3 parts work together. And it wasn't as simple as just a guide and a weapon. Nope, there was a secret handshake involved.
This right here was the moment of truth, the pivotal experiment. When the team mixed the Cas9 scissors and the crRNA guide with the target DNA, absolutely nothing happened. The scissors were just sitting there. But then they added that mysterious third piece, the tracrRNA, and bam, the DNA was sliced perfectly. That hidden accomplice wasn't just helpful, it was absolutely essential.
So they had cracked the code. Here's how it works. First, that tracrRNA has to find and pair up with the crRNA guide, forming this kind of dual-RNA structure. This structure then acts like a key, turning in an ignition. It flips a switch and activates the Cas9 protein. And only then can the guide RNA steer the now activated Cas9 to the target DNA and snip. A perfect, clean cut that completely disables the virus.
And they found out the cut itself was incredibly elegant. The Cas9 protein isn't just a clumsy axe, it's a precision tool. It has 2 separate cutting blades, these things called nuclease domains. One domain, HNH, cuts one strand of the DNA double helix, while the other, RuvC, cuts the other strand. It's a true set of molecular scissors making a perfect break. Now, just solving that mystery would have been a huge discovery, a fantastic piece of science. But this is where the story pivots. It goes from pure biology to world-changing engineering. See, the team wasn't just happy knowing how it worked. They immediately saw its mind-blowing potential. And they had this incredible what-if moment. This dual-RNA system was cool, but for using it in the lab, it was still a little complicated. So they asked a brilliant question. What if we could make this simpler? What if we could stitch the guide and the activator together into one single elegant molecule? And that's exactly what they did. Their solution was the single guide RNA.
They basically engineered a brand new molecule that fused the important parts of the cRNA and the tracrRNA into one piece. And just like that, instead of needing 3 components, a scientist only needed 2, the Cas9 scissors and this single, easy-to-create, totally programmable guide. This was the masterstroke.
They knew they were onto something massive. In the paper's conclusion, they wrote what has to be one of the greatest understatements in the history of science. They said their system could offer considerable potential for editing genomes. Let me tell you, considerable didn't even begin to cover it.
In that brilliant leap, simplifying the natural system into an easy-to-use 2-part tool is precisely why this single paper launched a full-blown revolution in biology.
What this discovery did was give the world a tool that wasn't just powerful, it was also cheap, it was efficient, and it was ridiculously easy to program. By turning nature's complex defense into a simple tool, it became accessible to practically any molecular biology lab on the planet. It completely democratized gene editing. It changed the game for basic research and, for the first time, opened a real, tangible path toward new therapies for thousands of genetic diseases.
So think about it. A journey that started by asking a simple question about how bacteria fight viruses ended up giving humanity the power to rewrite the code of life itself. A wonder of nature became a world-changing technology, and it leaves all of us with maybe the most profound question of our time. Now that we can, what will we choose to write in the book of life?